Thank you, Lang, and thanks, Thomas, for the introduction. I do indeed recommend the book. I couldn't say otherwise. Thank you all for being here. I'm amazed after all the dancing you did last night, you look all fresh and alert, and I'll try and keep you that way. I know I'm the last thing between your coffee and, well, or rather, I'm the last thing between you and your coffee, so I'll try and be concise. My talk today couldn't be more different than the last talk. Thomas showed us some beautiful mechanistically based science, deep science-based calculations that we can use to make our nanotoxicology more meaningful. I'm showing you everything I'm showing you today is empirical, and that's not because I don't believe in calculations. In fact, I do have projects involving a lot of computational calculation modeling going on at the moment, but today the day is for NanoRetox as far as I'm concerned, a 4-year project we just finished. And this is a very long list of names of all my collaborators who I would start and finish by acknowledging, because this project wouldn't have been possible without them, and it has been indeed a team effort. And I hope I'll show you that with my next few slides. Guessing this is how this works? Yes, fantastic. Okay, so just some boring basic statistics, and please forgive the animations. I didn't intentionally put them there, so they look a bit weird. So NanoRetox finished in December. It was a small, medium-scale FP7 funded collaborative project. The word collaborative should probably be in capital. Lasted 4 years, and at the inception point, when we wrote that proposal, which is perhaps more than 5 years ago now, our main objective was how broad and how naive, to identify the potential risks to the environment and human health posed by free manufactured nanomaterials. I think we write far more elegant proposals these days, but I think the value of this work is really the amount of data we collected, and I hope I'll show it to you here today. So we had 3 main strands to the project. The one was that we would do everything from making the particles, characterizing them, labeling them, testing how reactive they are, because one of the things we wanted to check was whether we could predict, very much along the lines of what Thomas said earlier, use properties, physical chemical properties, to predict toxicity. We also did a lot of ecotoxicology, and that was another key strand to our project. We didn't want, at the time, the human toxicology, perhaps even still these days, was a lot more popular and a lot more well-developed. Ecotoxicology was the poor relative, so we gave it center stage in our project. And of course, we felt that human toxicology was also important, and it had to be a component to what we were doing. So for each one of these strands, what we thought were our special project strengths was that, as I said, indeed, we produced every single particle we tested ourselves, perhaps with just one exception. And when I say ourselves, we had also industry partners. So some of our particles were more realistic, industrially relevant, but they were produced by our own industrial partners, following the spirit of the project, if you like, not necessarily always producing perfectly beautiful monodispersed fantastic particles like we did in the lab, but really particles that tried to complement the rest of our database. And we worked very hard to ensure quality and reproducibility throughout the project. So in fact, we tested multiple batches of the same particles, and we can confidently say that multiple syntheses give the same toxicity results, so that gives us some confidence in our methods. We did stable isotope labels uniquely. We were the first to develop stable isotope labeling, in fact, and we still are one of the few labs that can do that, and we do that for QNANO, incidentally, so that's an advert. Use us in QNANO if you want to see stable isotope labeling work for your samples, and it does. It's really a fantastic technique, particularly for ecotoxicology. What it gave us that others didn't have at the time was the ability to measure very, very low doses of nanoparticles and make the systems very realistic. And that's because tracing the isotope label is at orders of magnitude more sensitive, or can be orders of magnitude more sensitive than tracing unlabeled nanoparticles of zinc oxide, for example, which is everywhere. Our organisms had more zinc oxide in their bodies than we would dose them in our experiments. And I already said about characterization and reactivity, so I'll move on to my next point. The unique features our work had on ecotoxicology, and are all mixed up in my slides, were that we selected our organisms not as standard OECD organisms. Is Tom here? No, okay, I'm safe. I can say whatever I want. So we chose organisms we thought were either more relevant, more vulnerable, more good indicators of what nanotoxicity could bring to the environment. So we really worked with these organisms. You can see they're really ugly, but they did the job. I've mentioned stable isotope labels, and we also combined in vivo work with in vitro, and I'll go into human toxicology and make that point one more time. We did in vitro work on mammalian cell lines, and again we linked in vitro human toxicology with in vitro ecotoxicology. So the techniques were comparable. At the start of the project, we really felt that we would have to treat ecotoxicology and human toxicology as two totally different entities, as everybody else did. What we found amazingly was that, in fact, both our sets of experiments agree and give us the same answers. So if you harmonize, and you see some more of the harmonizing we did later, but not too much later, in 5 minutes perhaps. But if you harmonize your methodologies and your systems, you can actually get the same answers from both human and environmental toxicity experiments. We did some genotoxicity work. I'm not going to speak about that today. So this is what we tested. Please forgive the boring slides. I'm really just trying to give you a flavor of 4 years worth of work from 12 labs. I'm not going to show you beautiful diagrams, partly because a lot of this work is currently submitted for publication, and I don't want to preempt these publications. I really just want to give you the big picture of NanoRetox project and some flavor of what we did and how we have the confidence to make the statements I will be making in a while, which is because we have such a big database. So 8 classes of nanomaterials. Once you add all the variants, so different cappings, different synthesis method, different size, we had 55 single types of nanomaterials we tested in a whole range of different tests, as you can see, 33 in vitro tests, 23 in vivo tests, 11 single-dose tests, 8 species plus human cells. So a really broad range of data there that we can use now to give you, some generic answers, if you like. And these are our hypotheses. Now, I know a lot of people in this particular audience will be unhappy with my spelling. So where it says S-I-Y-E actually means size. So a lot of people will be unhappy with me saying size of particle does not affect toxicity. And in fact, we phrased those hypotheses we're testing intentionally to be controversial, and here's what influenced us. One of the things we did for NanoRetox project was science communication, and we arranged a meeting in London where we organized it. I know a couple of people in this audience were there with the Chatham House rules, which, to those of you who never heard of this before, these rules imply that when you go to this meeting, you can say whatever you like. This can be reported, but it can't be attributed to you. Nobody knew who said what. And that revealed some very, very interesting opinions from industry who seem to be very agreeable to the way I present my hypothesis here. So in fact, we chose to put the hypothesis in a way that we could refute them rather than in a way that we could prove them, and you will see that in a minute. You saw in my previous slide a whole range of hypotheses to do with how important agglomeration is, how important solubility is. I'm not going to talk about all this data, or I would need at least another 20 minutes, which I don't have. I chose a couple of hypotheses that I think our data show really nicely, and this is all I'm going to talk to you about today. So one of these was that we demonstrated that the nature of the test, or to say that the nature of the test is not important is actually a mistake, and it does really matter what test you choose to use in what answer you get. So we found that cell lines show different sensitivities, and I know many people won't think, okay, so what? Different species have different sensitivities, and when you bring all that together, then you start seeing the big picture of overall environmental or human sensitivity to nanoparticle exposure. We also found that the exposure routes we chose had a huge impact on the result. For example, diet versus water. And we also found that sublethal measures were very, very sensitive indicators. Now, there's no ecotoxicologist in this audience that won't say, oh yes, of course, we told you so a long time ago. So this is not a message for you guys, I know you know. This is a message for everybody else. Okay, so a little bit exploring this first hypothesis a bit more. We found that what's used, for example, by all OECD tests, the in vivo adult mortality, is the worst indicator you can choose to really see what impact the nanomaterials we're testing have to environmental indicators. We found, for example, that for snails and polychaetes, we didn't see any mortality really. So all the effects we saw were sublethal effects. For mussels, just one test gave us mortality, but we had 7 instances out of these various tests I showed you earlier, or rather I didn't show you, I mentioned we did. 7 actually did show sublethal responses. So if you were just looking at mortality, you would have missed these effects. Perhaps on a sort of mortality or a small mortality flag can be shown by zebrafish, which do actually seem to die quite sensitively when in relevant doses. And remember, I did emphasize all the work we did was on environmentally relevant doses. And zebrafish are reasonably sensitive, but no other organisms we tested. So really the big conclusions here are that do use other indicator tests because they will tell you more. Don't just stick to adult acute toxicity tests. And that we feel that perhaps OECD protocols should eventually include that type of tests and consideration of risk of evaluation of this type of risks. Now the other big discovery, if you like, we made through nanoretox was that manufactured nanomaterials can be more toxic than aqueous metals or can be less toxic than aqueous metals. Now, this would appear as a very mixed result. So perhaps some 17 out of however many tests showed that nano was less toxic than aqueous. Then some tests showed similar toxicity and some showed that nano were more toxic than aqueous. Is this just a spread in the data? Well, we think this means something. First of all, so that we don't miss this point, the manufactured nanomaterials we tested were all bioavailable, and that also means something. So we found them in our organisms when we fed them or when we watered them with spiked waters and food. So we did find that many of the standard toxicity tests actually showed the nanoparticles to be less toxic than an aqueous metal. And this, in fact, as I specifically write here, this is not different from what other people in the literature found, so that's not the big news. But what may be a bit more of big news, if you like, that we discovered that some tests conclusively, repeatedly, show the particles to be more toxic than aqueous. And these are specific types of tests, which if you don't do, and again, they are sublethal tests, so you miss this effect. For example, we found that when you feed the nanoparticles to the organism or through the sediment exposure, then you find the nanoparticles to impose more toxicity than aqueous. So particular tests and particular organisms as well. So this is really just I'm just summarizing this point in this slide. So I'll move to my next point, which is that just because a particle is nano, that doesn't make a particle toxic. Okay, so perhaps that's not what we want to say. Perhaps people are very keen to emphasize the nano, but it's actually what we found was not. The nano itself is not the toxicant. So here's two sets of results. We did a huge number of tests with silica nanoparticles, different sizes, different shapes, different sources. Not much toxicity. Very nano, but not toxic. So being nano doesn't make you toxic. Same for titania. Titania did show a little bit of moderate toxicity in some instances, but nothing too drastic. Again, if you're made of TiO2, you're not very toxic compared to other things, however small you are. Perhaps one of our big findings, and what I want you to take from this talk, is that what we managed to refute is that you are indeed, or we refuted that you cannot rank the toxicity on the basis of composition, and my picture here shows exactly that. Now, again, it would take me another whole talk to explain to you what this graph shows, but it shows you on the vertical axis we have a whole range of different tests from different labs, and this is a heat map, okay? So red means toxic, green means non-toxic, yellow shows something in between. So you will see that, as I said in my previous slide, if you're made of titania or silica, you're not toxic. You might be a tiny bit annoying, but nothing too bad. The worst toxicant from these sets of nanoparticles we tested, and the most consistent toxicant of all, was copper oxide, actually, perhaps surprisingly not silver, as we and perhaps others would have expected. Silver is an amazingly mixed bag, and you will see it's the only nanoparticle in this heat map that shows both reds and greens. It's we think we understand why, and I don't think this is the time to make a very long and complicated explanation. This is just to show you the breadth of our results, which allows us to make these big, big statements, if you like. So we've been working with this is the only graph I'm showing you today, and very close to the end of my talk, Lang will be happy to hear. So we tried to translate our data in some more graphic form, just to make the point that, for example, you can see, you can see this guys show low toxicity. So if this is your relative toxicity, very high, medium, low. These are the bad guys, if you like. And look at silver, look at the spread. So some things are consistently very highly toxic. Silver can be anything. And there's a good number of reasons why that is, and I won't tell you now. So really, what am I going to tell you now? I'll skip the first couple of conclusions. These are conclusions from the whole project. I really didn't show you data to support these conclusions, but I want you to take home the message that chemical composition seems to be the primary variant. It doesn't matter how many different sizes of silver. I can see Thomas shaking his head. I'm glad we agree. It doesn't matter how many different types of silver you test, you will find it more toxic than titanium, and so composition is everything. Size matters within your range of toxicity, but not beyond that range. And I would also like to emphasize that yes, we did see a nano effect. It's specific, it's clear, it's separate from aqueous toxicity. And we think we know why. Thank you again to my wonderful Nano-Redox Consortium, and thank you to you all. And questions, please.
Thank you, Eva. Well, I'm going to delay your coffee break by 2 minutes by giving Eva a chance to answer at least one question. I'm sure there must be a question from the audience.
Thank you. It looks like that the project generated a lot of data, and you gave some conclusions. You looked at a number of hypotheses, single factors. Did you actually also look at correlation between those factors, that they might actually have opposite effects and actually affect each other, as in a multivariate response pattern?
We did, is the brief and easy answer, because we were able to check each variable in more than one sets of particles. We could exclude, for example, test size and shape, or size and surface modification, solubility, and these. So because we could test the same variable in more than one systems, we could then say that it's not the other one, it's this one. If I'm sorry, I'm not making a very good point, but the answer, I think, is yes, and we can discuss this more over coffee.
Okay. Thank you, Eva. Thank you so much.